Welcome to Lecture 8.2, in which we will go over eukaryotic cell division. Our learning objectives for this video are to describe the levels of organization for eukaryotic DNA, define ploidy and homologous chromosomes, describe how eukaryotic organisms divide through the cell cycle and distinguish between its different phases, define the G0 phase, and continue learning about the similarities and differences between binary fission, mitosis, and meiosis. The eukaryotic genome is contained in multiple linear double-stranded DNA, instead of a single circular double-stranded DNA. The DNA is condensed to fit inside the nucleus by being wrapped around nucleosomes, which are complexes made of proteins called histones. We will talk a bit more about histones and how they affect the way genes are expressed in the epigenetics lecture. DNA wrapped around nucleosomes is often referred to as beads on a string. The DNA wrapped around nucleosomes is further folded and compacted into structures called chromatin. The chromatin is then further compacted into chromosomes. The word ploidy refers to the number of copies of each chromosome an organism has. A haploid is an organism that has just one set of each chromosome. A diploid is an organism that has two sets of each chromosome, or in other words, one pair of each chromosome. Some eukaryotes are completely haploid, while others are generally diploid but have haploid life stages, or vice versa. Humans, for example, are diploid but have reproductive cells that are haploid. A karyotype refers to the complete set of chromosomes from an individual. This is a karyotype of a male human. Humans have 23 pairs of chromosomes, for 46 total. One is a sex chromosome. Males have one X chromosome and one Y chromosome that pair up, while females have two X chromosomes. Since diploids have two copies of each chromosome, each pair is referred to as homologous chromosomes. Homo means same, referring to how these chromosomes contain the same genes in the same locations. We will learn more about this in meiosis, and in the genetics lecture, we will learn about how genes on each chromosome can have slight variations. The cell cycle is a series of events that occur in a specific order and result in one cell growing and dividing into two cells. The cell cycle can be divided into two major phases, which can themselves be divided into multiple phases. First is interphase, and then the mitotic phase. Interphase is when the cells grow and replicate their DNA. As the diagram shows, it makes up the majority of the time spent in the cell cycle. It can be further divided into the G1, S, and G2 phase. The G1 phase stands for gap or growth phase 1. This refers to how it's the phase that fills in the gap between the cell finishing mitosis and starting the cell cycle again, or how the cell spends this phase accumulating energy and building blocks so that it can grow. The building blocks are important for making the proteins needed for DNA replication and cell division. Generally, the G1 phase is the longest phase of the cell cycle and where many dividing cells spend their time. The S phase stands for the synthesis phase. This refers to how it's the phase in which another copy of DNA is synthesized. In other words, DNA replication of each chromosome occurs at this step. This image shows a pair of homologous chromosomes being replicated. Remember that in humans, there would be 23 pairs. The different colors represent how each chromosome comes from a different parent. Over the course of S phase, the chromosomes are duplicated. The two identical copies of a single chromosome are referred to as sister chromatids, and they are connected by the centromere in the center. While we often think of chromosomes as being X-shaped, it is really only after they've been duplicated that they have that appearance. The G2 phase stands for gap or growth phase 2. At this point, all the chromosomes should be completely duplicated. This is another phase for preparing and sometimes also further growth. The cell begins making proteins that will allow it to separate the chromosomes into each cell later on. Some organelles, such as the centrioles, are also duplicated. The cytoskeleton gets disassembled so that the tubulin can be used for mitosis. Mitosis is, overall, the process in which replicated chromosomes align and then separate so that the DNA and other cell components are equally distributed between the two resulting cells. Mitosis itself can be divided into several phases, and cytokinesis is considered a separate phase, although it sometimes occurs with the end of mitosis. This diagram shows both a schematic and actual microscopy images for each phase. The tubulin that was broken down from the cytoskeleton is used to form the mitotic spindles that are in green in the microscope images, and the DNA is in blue. The first step of mitosis is prophase. During interphase, the chromosomes were actually not as condensed because they were being replicated. Towards the end of G2 and during prophase, the chromosomes truly condense and can even be seen on a light microscope. The mitotic spindle also begins to form. The purpose of mitotic spindles is to attach to the chromosomes and separate the sister chromatids from one another. During this time, the nuclear envelope also breaks down, represented by the holes in the nuclear envelope. Prometaphase is essentially named for being the phase between prophase and metaphase. It starts after the nuclear envelope is completely disassembled. During this phase, the mitotic spindles attach to the kinetochores, which are proteins found on each side of the centromere that allow for spindle attachment. The centromeres themselves are still holding the sister chromatids together. Metaphase is the alignment phase of mitosis. During metaphase, the chromosomes begin to align in the center of the two forming cells. Where they align is referred to as the metaphase plate, represented by this dashed line. The metaphase plate is not a physical structure, and is just the region where the chromosomes line up. Anaphase is the separation phase of mitosis. This is when the sister chromatids are pulled apart. Once pulled apart, the chromosomes are pulled towards the opposite ends, or poles, of the developing cells. The pulling is done by the mitotic spindle shortening. At the same time, the cell elongates as the poles physically move apart from one another. Telophase is the final phase of mitosis. In a way, it's the opposite of what happens in prophase. Once the chromosomes reach their respective opposite ends, they unravel and become less condensed. The mitotic spindles are broken down so that the cytoskeleton can reform, and the nuclear envelope also begins to reform. Cytokinesis is the phase in which the two cells are actually physically separated from one another, allowing for cell division to completely finish. The way it occurs is different in animals and plants. In animals, cytokinesis actually begins as anaphase ends and occurs with telophase. A cleavage furrow is formed, and it pinches off to form two cells. Plant cells have rigid cell walls, so they are unable to make cleavage furrows. Instead, they form a cell plate using Golgi vesicles, and these eventually merge to form a new cell wall that divides the two new plant cells. Different cells divide at different rates, and some aren't very active in dividing at all. Cells that are not actively dividing are said to be in the G0 phase. Sometimes cells enter this phase temporarily, until a signal comes along that tells them to begin the G1 phase. Sometimes cells just stay in the G0 phase and stop dividing entirely. Some cells that divide quickly, such as skin cells and the cells that line the intestine, do not stay in this phase for long. Cells like mature neurons generally stay in the G0 phase and do little to no cell division. In this video, we learned that DNA is wrapped around nucleosomes, which are composed of histone proteins, and then further condensed into chromatin and chromosomes. We learned about ploidy and how haploid means having one copy of each chromosome, and diploid means having two copies of each chromosome. The cell cycle can be divided into interphase and the mitotic phase, with interphase divided into the G1, S, and G2 phase, and mitosis divided into prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis occurs at the end of cell division and fully separates the cells. Cells that are not actively dividing are in the G0 phase. In the next video, we will learn how the cell cycle is regulated. Thanks for watching.